Okay, so this year's Nobel Prize in chemistry has really got people talking, and with good reason. I mean, we're talking about like the very building blocks of life here, proteins.
It is remarkable.
Yeah.
It really is.
Yeah.
The committee recognized not one, but two incredible breakthroughs.
Right.
It's like they opened two doors to the same room from completely different sides.
It's like a 1-2 punch to this major scientific challenge.
Absolutely.
On one hand, we've got David Baker,
Yeah.
who's figured out how to design new proteins like from scratch.
Incredible.
And on the other, Demis Hassabis and Jumper, who've developed AlphaFold 2,
Amazing.
which is an AI that can predict how any protein will fold.
It's pretty wild when you think about it. It's like we've gone from barely being able to read the language of proteins to now being able to write it.
Oh, wow.
And predict it with amazing accuracy.
So before we get lost in the details, can you remind me why this is such a big deal?
Sure.
For someone like me who doesn't spend all day thinking about proteins?
So proteins are the real workhorses in our bodies.
Okay.
And in pretty much all living things, they do almost everything.
Okay.
From digesting your food to fighting off infections to making your muscles move.
Wow.
So the better we understand them, the better we can understand how life itself works.
Right.
And how to manipulate it for our benefit.
So it's not just about understanding these tiny little things, it's about understanding what they do.
Precisely.
Okay, I'm starting to see the potential here.
Yeah, and what makes this year's prize so exciting is that it's not just about understanding existing proteins.
Right.
Now we can design new proteins with specific functions.
Oh, wow.
And predict how any protein will fold into its 3D shape. That opens up possibilities we could only dream of before.
Okay, hold on, let's back up for a second. David Baker designing proteins.
Yeah.
That sounds like something straight out of science fiction.
It's really incredible.
How does he actually do that?
So he uses computer models to simulate how different amino acids, the building blocks of proteins, will interact with each other.
Uh-huh.
It's kind of like playing with Legos.
Okay.
But instead of building a castle, you're building a protein.
Yeah.
With a very specific function in mind.
So he's essentially building a protein blueprint on a computer.
A blueprint or a code.
And then how do you actually make a protein from a digital blueprint?
So that blueprint tells scientists the precise sequence of amino acids needed.
Okay.
And then they can synthesize the protein in a lab.
Got it.
And test whether it behaves the way they intended.
Wow. So it's not just theoretical. He's actually building these new proteins in the real world.
Yeah, it's remarkable.
What kind of things are we talking about? Like, what can these designer proteins actually do?
So imagine you're trying to treat a disease like cancer.
Yeah.
What if we could design a protein that specifically targets and destroys cancer cells without harming healthy tissue?
That would be incredible.
Much better than chemotherapy.
Yeah, way better than chemo, which basically attacks your whole body.
Exactly. And that's just one example. We could design proteins that break down pollutants in the environment or create new materials with incredible strength and flexibility based on protein structures.
Okay, my mind is officially blown.
It's really quite something.
It's like this research gives us a whole new toolkit to tackle problems we've been struggling with for years.
Absolutely.
But you mentioned another side to this year's prize, right? Something about predicting how proteins fold.
Yeah. So this was a huge problem for decades.
Yeah.
Predicting how a protein will fold into its 3D shape, which determines its function, was incredibly difficult.
Yeah.
It's like knowing all the letters in a word, but not knowing how to arrange them to make a meaningful sentence.
So how did they finally solve it?
That's where AlphaFold 2 comes in.
Okay.
It's an AI system developed by Demis Hassabis and Jumper.
Right.
It's basically a supercomputer that has learned the intricate rules of protein folding by analyzing a massive database of known protein structures.
So it's like they gave this AI a protein origami manual and let it practice for years until it became a master folder.
That's a great way to put it.
Wow.
And now AlphaFold 2 can predict the 3D structure of almost any protein with remarkable accuracy.
Really?
Just from its amino acid sequence.
Okay, I see where this is going. Now we can design new proteins and then use this AI to double check if our designs will actually work.
Precisely. And that's just one way they work together.
Wow.
AlphaFold 2 can also help us understand existing proteins in more detail.
Okay.
Potentially leading to new diagnostic tools.
Right.
Or even therapies for genetic diseases.
Okay, so we've got these two amazing tools, but where do we go from here?
That's where things get really interesting.
Okay.
Because we're not just talking about solving individual problems. We're talking about a fundamental shift in how we approach science and technology.
Wow.
Imagine a future where we can design personalized medicine tailored to your specific genetic makeup, or create sustainable materials inspired by the unique properties of proteins.
It's like we're on the cusp of a whole new era.
We are, and it's not just about specific applications, it's about a fundamental shift in how we approach science and technology. With these tools, we're not just observing and studying nature.
Right.
We're becoming active participants in shaping it. That's
a pretty powerful thought. So we're not just understanding proteins, we're like learning to speak their language, to write it even.
Exactly. And that's what's so exciting about the combination of Baker's work and AlphaFold 2. We can design a protein on a computer and then use AlphaFold 2 to see how that design will actually fold in the real world, to make sure it's going to function the way we want it to.
It's like having a built-in quality control system.
Yeah.
You can basically test your protein design in a virtual world before you even go into the lab.
Precisely. And that saves enormous amounts of time and resources. We can focus on the most promising designs without having to go through, countless trial and error experiments.
So it's not just about the science itself, it's about accelerating the pace of discovery. We can test ideas and iterate much faster than we could before.
Absolutely. And think about all the potential applications. We've talked about medicine and material science, but there's so much more: agriculture, energy production, environmental remediation. The possibilities are truly vast.
Yeah, it's almost hard to wrap your head around all the potential, but I keep coming back to this idea of speaking the language of proteins. What else can we learn from this new level of understanding?
Well, AlphaFold 2 isn't just about designing new proteins. It can also help us understand existing proteins in much greater detail. Think of it like having a detailed map of the protein universe.
Okay, I'm following you, so we can see how all these proteins are connected, how they interact with each other, how they function in different environments.
Exactly. We can use this information to understand how proteins evolved, how they work together in complex biological systems, and how mutations can affect their structure and lead to diseases.
So this could lead to new insights into genetic diseases and potentially even new treatments.
Precisely. Imagine being able to predict how a specific genetic mutation will alter the structure of a protein, and then design a drug that corrects that structural defect. That's personalized medicine taken to a whole new level.
Okay, now we're getting into some seriously futuristic stuff. It's almost like we're rewriting the rules of biology.
In a way, we're moving from a passive understanding of biology to an active one, where we can design and engineer biological systems with unprecedented precision.
This is incredible, but I have to ask, where does it all end? I mean, if we can design proteins to do practically anything, what are the limits?
That's a great question, and one that we need to be thinking about very carefully. Because as with any powerful technology, there are potential risks and ethical considerations that we need to address.
That makes sense. We can't just get caught up in the excitement of all this new potential without thinking about the bigger picture.
Exactly. So while we marvel at the possibilities, we also need to be asking ourselves some tough questions about how we use this newfound power responsibly. So while we marvel at the possibilities, we also need to be asking ourselves some tough questions about how we use this newfound power responsibly.
It feels like we're entering like a whole new world almost. It's exciting and a little bit daunting at the same time.
Yeah, I agree. It's a bit like exploring a vast uncharted territory. There's so much potential for discovery and innovation, but we need to proceed with caution and a deep sense of responsibility.
So where do we even begin to address these concerns? It feels like we're talking about something much bigger than just science.
You're right. This goes beyond the realm of science and touches on fundamental questions about what it means to be human and how we want to shape the future of life itself.
Okay, so it's not just about the technical aspects of protein engineering, it's about the ethical and societal implications as well.
Yes, exactly. We need to have open and honest conversations about the potential risks and benefits of these technologies, and we need to involve a wide range of perspectives: scientists, ethicists, policymakers, and the public at large.
That makes sense. It can't just be a small group of experts making decisions that could affect all of us.
Right. We need to democratize these conversations and ensure that everyone has a voice in shaping this future, because ultimately it's about the kind of world we want to create for ourselves and future generations.
So what can we do as individuals? I mean, it's easy to feel overwhelmed by the scale of these challenges.
Well, for starters, we can educate ourselves about these technologies and their potential implications. The more we understand the science, the better equipped we are to engage in informed discussions and make responsible choices.
That's a good point. Knowledge is power, as they say, but education is just the first step, right? What else can we do to ensure that these technologies are used for good?
We can support research and development that prioritizes ethical considerations. We can advocate for policies that promote transparency and accountability in the field of protein engineering. And we can engage in constructive dialogue with our communities and elected officials about the future we want to see.
So it's about taking action on multiple levels, individual, community, and societal. It's about making sure that these powerful tools are used wisely and for the benefit of all.
Exactly. It's about shaping a future where protein engineering serves humanity and the planet rather than the other way around.
This deep dive has really given me a lot to think about. I came in excited about the science, but now I'm realizing that there's a whole other layer of complexity that we need to grapple with.
That's the thing about scientific breakthroughs. They often force us to confront not just the how, but also the why. In the case of protein engineering, the why is perhaps even more important than the how.
Because we're not just talking about building things, we're talking about shaping the future of life itself.
Precisely, and that's a responsibility we can't take lightly.
Well said, and to our listeners, thank you for joining us on this deep dive into the world of proteins. We hope you've enjoyed the journey and that you'll continue to explore these fascinating and complex issues with us. Until next time, stay curious,